Welcome to iMODE-CKD. iMODE-CKD integrates multidisciplinary expertise from leading European academic and industrial investigators in the form of a unique training platform focusing on biomarker research and systems biology on chronic kidney disease, or CKD. Chronic kidney disease is a rising global health problem. It is caused when the kidneys lose their function over time. High blood sugar or high blood pressure are the two most common causes of CKD and are associated with a high risk of death from cardiovascular disease. Pathologies in parts of the kidney, called glomerulus, are causing the glomerulopathies, which are also leading to CKD. It is irreversible, hard to detect at an early stage, and, during the final stage, the kidney loses complete function, leaving two options: transplantation or hemodialysis. In order to study chronic kidney disease, a total of 12 academic partners and 3 industrial partners are joining forces in the iMODE-CKD project, with a special emphasis on the application of a wide range of omics and bioinformatics techniques within clinical research. iMODE-CKD provides the unique opportunity to train young researchers in the different fields of omics research and also in omics data evaluation, combining datasets from different omics traits and exploring both data and literature to enable the generation of in silico models of the molecular pathology of specific diseases. But how will iMODE-CKD study chronic kidney disease? There are a number of independent projects focusing on one purpose: understanding chronic kidney disease and provide the tools to help handle the disease. The Department of Nephrology in the University Clinical Centre of Skopje has long-standing activities in clinical research in vascular calcification, kidney biopsy registry, and kidney transplant medicine. From 2011, the centre is leading the CKD bio-study on the progression of CKD in various types of primary glomerulonephritis. In the CKD bio-study, clinical samples collected annually and are available for further evaluation by the respective collaborators and partners. Within the iMODE-CKD project, the expectations are to obtain a definition of the clinical core parameter set, unify clinical databases for CKD, and evaluate biomarkers in the context of clinical data, focusing on CKD progression and cardiovascular disease complications. The Biomedical Research Foundation of the Academy of Athens is a non-profit institute dedicated to understanding, treating and preventing human ailments through biomedical research. The Biotechnology Department has successfully described the application of various multidimensional approaches for the comprehensive analysis of the urinary proteome and the identification of disease biomarkers using state-of-the-art equipment such as MALDI-TOF, LC-MALDI and LC-MSMS Orbitrap. In the iMODE-CKD project, the urine proteome from CKD patients will be analysed in order to provide an initial list of differentially expressed proteins, followed by functional annotation and prioritization of findings, and then the verification of selected proteins by complementary assays. Mosaics Diagnostics is the leading specialist in clinical proteomics and capillary electrophoresis coupled to mass spectrometry analysis. Research activities involve the identification of urinary biomarkers for human diseases, sequencing of peptide biomarkers, and the development of software to analyze multidimensional data. In the iMODE-CKD project, CEMS will be employed to investigate human urine samples from patients, aiming at the identification of biomarkers specific for detecting the progression of primary glomerulonephritis and the assessment of the value of these identified biomarkers to predict CKD progression. In the renal fibrosis lab in Toulouse, part of the French National Institute of Health, the research is focused on the understanding of the mechanisms of early fibrogenic stages, on the transformation of this knowledge into therapeutic targets, and on the identification of early, non-invasive biomarkers of renal fibrosis. In the iMODE-CKD project, microRNA modifications in urine from CKD patients will be studied by using next-generation sequencing technologies, real-time PCR, and available web tools, with the final objective being to identify differentially expressed microRNAs involved in CKD pathology. The Institute for Molecular Cardiovascular Research has strong expertise in chromatographic techniques, working on the isolation, identification, and physiological characterization of endogenous cardiovascular mediators from cell secretomes, serum, and plasma in order to identify peptides, proteins, and nucleotides. In the context of the iMODE-CKD project, plasma peptides and proteins and their post-translational modifications will be identified and quantified using LC electrospray MS, MALDI FT Orbitrap, and MALDI TOF MS. And finally, the impact of specific post-translational modifications of proteins and peptides on the affinity of uremic toxins will be described. The Department of Health Sciences at the University of Milano Bicocca is home to a highly experienced group of researchers with multidisciplinary expertise that are applied to a number of proteomic and lipidomic studies, including the molecular characterization of renal cell carcinoma and diabetic nephropathy. At UniMib, the proteomics laboratory is equipped with all the tools necessary for high-standard, high-throughput mass spectrometric analysis. As part of the iMODE-CKD project, MALDI MSI will be performed on fresh frozen and FFP renal biopsies in order to identify putative biomarkers indicative of CKD progression and to determine their spatial distribution within renal tissue, in accordance with a centralized histological diagnosis of all patients admitted at San Gerardo Hospital in Monza. Service XS is a leading-edge genomics and transcriptomics service provider in the area of molecular genetics. The company's main activities are next-generation sequencing, gene expression profiling, genotyping, and methylation analysis with access to Illumina, ion proton, and state-of-the-art microarray platforms. For the purpose of the iMODE-CKD project, the next-generation sequencing facilities will be used for expression profiling of tissue samples of cases and matched controls obtained in the context of this study, in order to find patterns in gene expression related to CKD and to determine splicing isoforms or somatic variants that might be associated with the kidney phenotype. The Analytical Biogeochemistry Department is working on targeted and non-targeted metabolomics of body fluids and on the identification and validation of new biomarkers. The work performed in the iMODE-CKD project will be used to generate metabolic master maps of plasma and urine during CKD progression using multidimensional chromatography in combination with ultra-high resolution 12 Tesla ICR FTMS and selected NMR analysis. The results will include new standard operating procedures for sample preparation and analysis, new small molecules indicative of treatment benefit, structural information on the potential of new biomarker metabolites, and finally, value assessment of the new biomarkers for diagnostics and to predict treatment response and monitoring. The Biomarkers and Systems Medicine Group at the University of Glasgow performs bioinformatics analysis and integration of high-throughput omics datasets. The contribution to the iMODE-CKD project will be to collect the progressively consolidated existing public data in collaboration with the BRFAA and perform analysis of the existing and de novo generated data from partners within the project. In the end, all the analysis will lead to the creation of the first molecular map of established CKD and processes indicative of progression and iterative modeling of processes utilizing profiling influx from the other partners, which will then be expanded to cover the various stages of CKD progression. The Computational Biology and Medicine Group will contribute to the iMODE-CKD project by extracting and analyzing publicly available and proprietary datasets and key publications relevant to CKD progression. Available datasets to be considered for integration, multi-source omics data collection platforms, and existing knowledge databases will be identified during the project and at the end create an omics database to be available for further statistical analysis for the identification of CKD progression associated clusters. Systems biology approaches will be utilized to combine the various omics datasets and reduce the biological, technological, and computational challenges of integrating complex data. This approach has the potential of not only aiding CKD biomarker and therapeutic target discovery, but also hypothesis testing for CKD clinical phenotype segmentation and patient stratification. Combining all of this knowledge, we anticipate identifying and validating molecules involved in the progression of renal and cardiovascular complications in established CKD through the use of clinical omics, and to also understand the molecular determinants of established CKD through integrative systems biology. We expect our results to contribute to the prognosis, diagnosis, and monitoring of chronic kidney disease with the employment of friendly and non-invasive tools. In turn, we hope that this can help reduce the increasing number of cases, improve the quality of life of the patients, offer a better future for the next generation, and to also diminish the severe health and economic burden imposed by this disease throughout Europe.